Agarose gel electrophoresis of PCR products. Agarose gel electrophoresis is a widely used technique for the separation and analysis of nucleic acids, particularly PCR, polymerase chain reaction, products. This method allows researchers to visualize DNA fragments based on their size, providing critical information regarding the success of PCR amplification and the presence of specific DNA sequences. Principle: Agarose is a polysaccharide derived from seaweed, which forms a gel when dissolved in buffer and cooled. The concentration of agarose in the gel can be adjusted to optimize the separation of DNA fragments of varying sizes. When an electric current is applied, negatively charged DNA fragments migrate towards the positive electrode. Smaller fragments move faster through the gel matrix than larger ones, allowing for size-based separation. Procedure: 1. Preparation of agarose gel. Agarose powder is mixed with a buffer solution, commonly TAE or TBE, and heated until fully dissolved. The concentration of agarose typically ranges from 0.7% to 2%, depending on the size of the DNA fragments to be separated. The solution is poured into a gel casting tray and allowed to solidify. 2. Loading samples. PCR products are mixed with a loading dye, which helps visualize the sample during electrophoresis. The dye also increases the sample's density, ensuring it sinks into the gel's wells. Samples are carefully pipetted into the wells of the gel. 3. Running the gel. The gel is placed in an electrophoresis chamber filled with buffer, and an electric current is applied. The electrophoresis duration depends on the gel size and the desired resolution. 4. Visualization. After electrophoresis, the gel is stained with a DNA-binding dye, such as ethidium bromide or SYBR green, which fluoresces under UV light to visualize the DNA bands. 5. Analysis. The DNA bands are compared against a DNA ladder, a mixture of known DNA sizes, to determine the sizes of the PCR products. This comparison helps confirm the presence of the target DNA sequence. Applications. Agarose gel electrophoresis is crucial for various applications, including verification of PCR products, ensuring the correct amplification of target sequences; restriction fragment length polymorphism (RFLP) analysis, analyzing genetic variations; cloning and sequencing, preparing DNA fragments for further molecular biology techniques. Detailed overview of Agarose gel electrophoresis of PCR products. 1. Gel preparation. Agarose concentration. The concentration of agarose in the gel determines the pore size, which affects the resolution of DNA separation. Common concentrations include 0.7%, suitable for separating larger fragments, greater than 1 kb. 1%, ideal for general applications, separating fragments from 500 bp to 10 kb. 1.5%, better for smaller fragments, 100 bp to 3 kb. 2%, used for very small fragments, less than 500 bp. Buffer choice. The buffer used during electrophoresis, TAE or TBE, provides ions that facilitate the conduction of electricity and maintain pH. TAE is often preferred for DNA analysis due to its compatibility with downstream applications, while TBE is used for high-resolution separations. 2. Sample preparation. PCR product cleanup. Before loading, PCR products may need purification to remove excess primers, nucleotides, and enzymes. Methods include column-based purification, using silica columns to bind DNA. Gel extraction, running the PCR product on a gel and extracting the band of interest. Loading dye. The loading dye typically contains a tracking dye, e.g., bromophenol blue, that migrates through the gel at a known rate, allowing researchers to monitor the progress of electrophoresis. Glycerol or sucrose is often included to increase sample density. 3. Electrophoresis setup. Gel casting. After the agarose solution cools to about 50 degrees Celsius, it is poured into a casting tray with a comb inserted to create wells. Once solidified, the comb is removed, leaving wells for sample loading. Electrophoresis chamber. The gel is placed in an electrophoresis tank filled with buffer. The electrodes are connected to a power supply, which applies a voltage across the gel. Typical voltages range from 80 to 150 volts, depending on the gel size and desired run time. 4. Running the gel. Monitoring migration. As the electric current is applied, DNA fragments begin to migrate towards the positive electrode, anode. The migration rate is influenced by size of DNA. Smaller fragments travel faster through the gel matrix. Shape of DNA. Linear DNA moves differently than circular or supercoiled DNA. Duration. The runtime can vary, but typically lasts 30 minutes to 2 hours. Researchers should monitor the dye front to avoid running off the gel. 5. Staining and visualization. Staining methods. Common DNA stains include ethidium bromide, intercalates between DNA bases and fluoresces under UV light. It is a potent mutagen, so safety precautions are necessary. SYBR green, a safer alternative that also binds to DNA and fluoresces, often used in real-time PCR. Visualization. After staining, the gel is placed on a UV transilluminator or a gel imaging system. The DNA bands appear as bright bands against a dark background, allowing for size estimation. 6. Analysis of results. DNA ladder. A DNA ladder, or marker, containing fragments of known sizes is run alongside the samples. By comparing the distance migrated by the PCR products to the ladder, the size of the amplified fragments can be estimated. Interpretation. The presence of a single band at the expected size indicates successful amplification of the target DNA. Multiple bands may suggest nonspecific amplification or primer-dimer formation. 7. Common troubleshooting issues. No bands. Possible reasons include failed PCR, incorrect annealing temperature, or degraded DNA. Smearing indicates overloading of samples, degraded DNA, or suboptimal gel concentration. Multiple bands suggests nonspecific amplification or primer-dimer artifacts. 8. Applications in research and diagnostics. Genotyping, used for analyzing genetic variations in populations. Mutation detection, identifying mutations in genes of interest. Cloning verification, confirming the presence and size of inserts in recombinant plasmids. Forensic analysis, analyzing DNA samples in criminal investigations. Detailed overview of errors in Agarose gel electrophoresis of PCR products. Agarose gel electrophoresis is a critical technique for analyzing PCR products, but errors can occur at various stages of the procedure. Understanding these potential pitfalls can help improve experimental outcomes and ensure reliable results. Below is a comprehensive breakdown of common errors and how to troubleshoot them. 1. Errors in gel preparation. Incorrect agarose concentration. Error: using an inappropriate agarose concentration can lead to poor resolution of DNA fragments. Impact: too low a concentration may not adequately separate large fragments, while too high a concentration can hinder the migration of larger fragments. Solution: choose the agarose concentration based on the expected size range of the PCR products, e.g., 0.7% for large fragments, 2% for small fragments. B. Incomplete dissolution of agarose. Error: not fully dissolving agarose can result in uneven gel consistency. Impact: this can cause inconsistencies in migration rates and band formation. Solution: ensure complete dissolution by heating the agarose solution thoroughly and swirling to mix. C. Air bubbles in the gel. Error: air bubbles can form during gel casting. Impact: bubbles can obstruct wells and lead to incomplete loading of samples. Solution: pour the agarose slowly and gently to minimize air incorporation, and use a comb that fits snugly in the gel tray. 2. Errors in sample preparation. Inadequate PCR product cleanup. Error: failing to purify PCR products before loading can lead to contamination with primers, nucleotides, or enzymes. Impact: this can result in extra bands or smearing in the gel. Solution: use a reliable purification method, such as column purification or gel extraction, to remove contaminants. B. Incorrect loading dye use. Error: using too much or too little loading dye can affect sample density and visibility. Impact: excess dye can obscure bands, while insufficient dye may cause samples to float out of the wells. Solution: follow recommended protocols for loading dye concentrations, typically around 10% of the total sample volume. C. Inconsistent sample volume. Error: loading different volumes of samples across lanes can lead to inaccurate comparisons. Impact: variability in band intensity makes it difficult to draw conclusions from the results. Solution: use a micropipette to load consistent volumes across all lanes. 3. Errors during electrophoresis. Incorrect voltage setting. Error: applying too high or too low voltage can affect migration rates. Impact: high voltage can cause band distortion or overheating, while low voltage can result in poor separation. Solution: use a voltage setting appropriate for the gel size and thickness, typically between 80 to 150 volts. B. Running time issues. Error: running the gel for too long or too short a time can lead to poor resolution. Impact: overrunning can cause bands to run off the gel, while underrunning may not achieve adequate separation. Solution: monitor the progress of the gel by observing the dye front and stop the run once the dye is near the bottom of the gel. C. Buffer depletion. Error: running the gel until the buffer is depleted can lead to inconsistent results. Impact: this can cause changes in pH and ionic strength, affecting DNA migration. Solution: ensure the buffer is filled to the appropriate level before starting electrophoresis and monitor during the run. 4. Errors in staining and visualization. Inadequate staining. Error: insufficient staining time or concentration can lead to faint or invisible bands. Impact: this makes it difficult to visualize and analyze PCR products. Solution: follow the manufacturer's instructions for staining protocols, ensuring adequate time for the dye to bind to the DNA. B. UV exposure issues. Error: prolonged exposure to UV light can damage DNA and lead to band degradation. Impact: this can result in smearing or loss of bands. Solution: minimize UV exposure time and use alternative imaging methods when possible, such as blue light transilluminators. C. Improper gel imaging. Error: using a poorly calibrated or malfunctioning imaging system. Impact: this can lead to inaccurate band intensity readings or misinterpretation of results. Solution: regularly calibrate imaging equipment and ensure it is functioning properly before use. 5. Errors in data analysis. Misinterpretation of bands. Error: incorrectly analyzing band patterns, such as mistaking primer dimers for target products. Impact: this can lead to false conclusions about PCR success or specificity. Solution: compare band sizes to a standard DNA ladder and consider running controls for accurate interpretation. B. Inconsistent controls. Error, not running appropriate positive and negative controls. Impact, this can lead to uncertainty about the validity of results. Solution, always include a positive control, known target, and a negative control, no DNA template, in every experiment. C. Failure to document results. Error, not adequately documenting gel images and analysis results. Impact, this can hinder reproducibility and the ability to reference results in the future. Solution, keep detailed records of gel images, annotations, and interpretations for future reference. Conclusion. Understanding and addressing these potential errors in Agarose gel electrophoresis of PCR products can significantly enhance the reliability and accuracy of molecular biology experiments. By following best practices and troubleshooting strategies, researchers can ensure that their results are valid and reproducible. Regular training and adherence to standardized protocols can further minimize the occurrence of these errors. 4. Errors in staining and visualization. Inadequate staining. Error, insufficient staining time or concentration can lead to faint or invisible bands. Impact, this makes it difficult to visualize and analyze PCR products. Solution, follow the manufacturer's instructions for staining protocols, ensuring adequate time for the dye to bind to the DNA. B. UV exposure issues. Error, prolonged exposure to UV light can damage DNA and lead to band degradation. Impact, this can result in smearing or loss of bands. Solution, minimize UV exposure time and use alternative imaging methods when possible, such as blue light transilluminators. C. Improper gel imaging. Error, using a poorly calibrated or malfunctioning imaging system. Impact, this can lead to inaccurate band intensity readings or misinterpretation of results. Solution, regularly calibrate imaging equipment and ensure it is functioning properly before use. 5. Errors in data analysis. Misinterpretation of bands. Error, incorrectly analyzing band patterns, such as mistaking primer dimers for target products. Impact, this can lead to false conclusions about PCR success or specificity. Solution, compare band sizes to a standard DNA ladder and consider running controls for accurate interpretation. B. Inconsistent controls. Conclusion. Understanding and addressing these potential errors in Agarose gel electrophoresis of PCR products can significantly enhance the reliability and accuracy of molecular biology experiments. By following best practices and troubleshooting strategies, researchers can ensure that their results are valid and reproducible. Regular training and adherence to standardized protocols can further minimize the occurrence of these errors. Detailed overview of errors in the investigation of figures from Agarose gel electrophoresis of PCR products. Analyzing the figures obtained from Agarose gel electrophoresis of PCR products is crucial for determining the success and specificity of the amplification process. However, various errors can arise during this analysis, leading to misinterpretation of results. Below, I detail common issues such as primer dimers, primer hairpins, RNA contamination, and other mistakes, along with references for further reading. 1. Primer dimers. Description: primer dimers occur when primers anneal to each other instead of the target DNA, forming short, unintended PCR products. Identification. Appearance: typically appear as small bands at the bottom of the gel, often below the expected size of the target product. Band intensity: may have higher intensity than the target band if the PCR conditions favor their formation. Causes: high primer concentration. Excessive amounts of primers can increase the likelihood of dimer formation. Low annealing temperature: a temperature that is too low can promote nonspecific binding. Solutions: optimize primer concentration. Use a lower concentration of primers. Increase annealing temperature. Adjust the annealing temperature to enhance specificity. References: Nolan, T. Hans, R.E., and Buston, S.A., 2006. Quantification of mRNA using real-time RT-PCR. Nature Protocols, 1, 3, 1559-1572.doi, 10.1038/nprop.2006.236. 2. Primer hairpins. Description: Hairpin structures form when the 3-foot end of a primer anneals to an upstream region of the same primer, creating a loop. Identification. Appearance may not produce a visible band on the gel or may appear as a faint band at an unexpected size. Impact on PCR can inhibit amplification by consuming primers. Causes. High complementarity. Primers with significant self-complementarity are more likely to form hairpins. Solutions. Design primers carefully. Use software tools to design primers with minimal self-complementarity. Check melting temperature, TM. Ensure TM values are appropriate for the specific PCR conditions. References. Kumar, A., and Kaur, M., 2015. Primer Design for PCR, a comparison of different software tools. International Journal of Current Microbiology and Applied Sciences, 4, 11, 126-135. 3. RNA contamination. Description: RNA may co-purify with DNA during the PCR process, leading to the appearance of unexpected bands. Identification. Appearance: bands corresponding to RNA often appear as high molecular weight bands or as smeared bands. Impact: RNA can interfere with downstream applications if not removed. Causes. Improper sample handling, inadequate separation or purification can lead to RNA contamination. Lack of RNase treatment, failure to treat samples with RNase can leave RNA intact. Solutions. Use RNase, treat samples with RNase to degrade RNA before PCR. Confirm DNA purity, use spectrophotometric methods to check for RNA contamination. 4. Nonspecific amplification. Description: nonspecific amplification occurs when primers bind to unintended sites, resulting in additional bands on the gel. Identification. Appearance: multiple bands at sizes different from the target product. Impact: this can complicate interpretation and reduce the specificity of the assay. Causes. Low specificity of primers. Primers may have low specificity due to mismatches with the target sequence. Inappropriate PCR conditions, suboptimal annealing temperatures or extension times can promote nonspecific binding. Solutions. Optimize PCR conditions, adjust the annealing temperature and extension time. Redesign primers, use software to redesign primers that are more specific to the target sequence. 5. Incomplete amplification. Description: this occurs when the PCR does not produce enough product, resulting in faint or absent bands. Identification. Appearance: very faint bands or no bands at all in the expected lane. Impact: low yield can hinder further analysis. Causes. Insufficient template: low amounts of DNA template can lead to weak amplification. Inhibitors in the reaction: contaminants in the sample can inhibit the PCR reaction. Solutions. Increase template amount, use more template DNA if feasible. Purify samples, ensure that samples are free from contaminants that could inhibit PCR. Conclusion. Errors in the investigation of figures from Agarose gel electrophoresis of PCR products can significantly impact the interpretation of results. By understanding these common issues, such as primer dimers, hairpins, RNA contamination, nonspecific amplification, incomplete amplification, and overloading, researchers can take preventative measures to ensure accurate analysis. Continuous education and adherence to best practices are essential for minimizing these errors and achieving reliable results.